we've been talking about biology, we've been talking generally about normal elements, normal atoms, and we've talked a little bit about ions. Remember, are atoms that have either gained or lost one or more electrons. Now let's talk about something else, which are isotopes. Isotopes are non-standard atoms that have extra neutrons. Now, this is important because you remember that the neutron does not have a charge associated with it. It's neutral. Protons have a positive charge, electrons have a negative charge. If there were extra protons in the nucleus, the element would have a different characteristic. It would have a greater positive charge. But when we change the number of neutrons, then we end up with the same element. It has the same characteristic because it has the same positive and negative charges, but it weighs more. It has more mass because remember, neutrons have actual mass, whereas electrons don't really have mass. We can add and subtract electrons. We don't really change any of the mass because they don't really weigh anything. But neutrons do have a lot of mass in relation to an atom. So we're going to talk about isotopes of carbon, but please remember that there are isotopes of many other elements that form naturally. Now, carbon makes two isotopes. Standard carbon is called carbon-12. 6 protons, 6 neutrons, 6 electrons, mass number 12. Carbon-13 has one extra neutron. It's called carbon-13. It has a mass number of 13, 6 protons, 7 neutrons, 6 electrons. Carbon-14, 6 protons, 8 neutrons, has a mass number of 14. Okay, same number of protons, same number of electrons, different number of neutrons. So the only thing that has changed is the mass. 99% of all the carbon on the Earth is carbon-12, just plain old normal carbon. 1% is carbon-13. It's rare, it's pretty stable, no big deal. A tiny, tiny amount, just a fraction of 1%, is carbon-14. Where did this stuff come from? All the carbon on Earth came from the sun. And we get new carbon on Earth in the form of gases from the sun, like carbon dioxide. And that new carbon in the carbon dioxide that flows to Earth from the sun has a teensy tiny less than 1% of the carbon in it is carbon-14. Now, carbon-14 is important because not only because it's heavier than regular carbon, but also because it's radioactive. Now, that's a big, scary word. You do not have to fear the carbon-14. There's not enough of it, and it's not radioactive enough to harm you. There are some things that are radioactive that will harm you, not carbon-14. Radioactive just means that it will decay, which means that it will give up one of these extra neutrons and go back to stable carbon-13, okay? All radioactive isotopes of all elements that have radioactive isotopes, all of them decay spontaneously, meaning that they don't have to be part of a reaction. They don't have to get hit by something. They don't have to, it just spontaneously, every once in a while, one of the neutrons is released from the atom. They also decay or give up those neutrons at a very predictable rate. And we can study this in the lab. We can measure this in the lab. We can take a sample of carbon, measure how many radioactive carbon-14 atoms are in it, and then measure the rate at which they shed neutrons and see how fast that radioactive decay happens. Okay, so we've measured this. We know exactly what it is. All radioactive isotopes have something that we call a half-life. The half-life is the time in which half of the existing amount has become non-radioactive. Okay, so if you have, let's say, 10 grams of C14 after 5,720 years. Hang on, I've got it somewhere. After about 5,000 years. 5 grams of it will no longer be carbon-14. Make sense? That's the half-life. All right, now, radioactivity, it sounds really scary. It can damage tissues, but it's really, really important in medicine. One way we use it is in something called a PET scan, positron emission tomography. You actually inject a patient with a radioactive substance and then put them in this big scanner to see how much of that radioactive sugar, is what they actually use, is being taken up by tissues in the body. And you can actually see if cancer has spread. You can detect the presence of cancerous tumors by seeing excessive sugar uptake in a PET scan. Okay, so that's one way that radioactivity can be used. X-rays use a different form of radioactivity, but it's still, an X-ray is a radioactive particle. Pass those through the body, they go more slowly, or they are absorbed more by hard substances such as bones, so you can get a very good picture of bones, not great pictures of soft tissues, but you can see where bones might be broken. You can see the placement of bones. This is the bones of the wrist and the hand. One of the other uses in science for isotopes is carbon dating, which is not two carbons going to dinner together, although that is cute, you gotta admit. Carbon-14, remember, is created by the sun. It's all new carbon-14 is always coming from the sun into the environment. Plants absorb carbon in the form of carbon dioxide and use that carbon dioxide plus water to make sugars. That's what photosynthesis is. How weird is it that plants make themselves out of water and air? How crazy is that? Okay, anyway. So you take carbon and water, put it together, make glucose and other sugars. That's what plants do. As they are bringing in new carbon dioxide and doing photosynthesis while they're alive, they're always taking in a teensy tiny itsy bitsy amount of carbon-14. We can then measure how much carbon-14 is left in something that's dead and figure out when it was alive and taking in new carbon-14. All right, so here's how we go. Carbon-14 arrives with normal carbon-12. Plants use the CO2 to make sugars. Some of that is carbon-14. When the plant dies, it stops taking in new carbon-14. And the amount of carbon that it had is 100%, like 100% of all the carbon-14 it's ever going to have. So animals eat the plants, they take in that carbon-12 and carbon-14. When the animal dies, or when the plant dies, it stops taking in new carbon. The tissues have 100% of the carbon-14 they will ever have. We know how much carbon-14 is in the general environment because it's been being sent from the sun. It is fairly stable, asterisk. Maybe it's not quite as stable as we thought. These might not be quite as precise as we thought. Again, it's always more complicated. This is still a good estimation. And then carbon-14 has a half-life of 5,700 years. See, I was really close. Which means that after 5,700 years, half of the carbon-14 is gone. So again, imagine that we had 10 grams after 5,700 years. 5 grams of that would still be carbon-14. Okay? So we can measure the ratio of carbon-14 to carbon-12 in formerly alive things and compare that to the half-life of carbon, and we can determine how long it has been since something died. Because if there's half the carbon that we would have expected at the beginning, then that thing is 5,700 years old. Half of the carbon-14 is gone. After another half-life, which gives us 11,400 years, another half is gone. So we halved the carbon-14 and then we half it again. So we have 25% left. Then after another 5,700 years, 17,100 years total, we have half again, 12.5%. Now you can see that if we keep going halvesies, 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 we're going to get to a point where we don't have any carbon-14 left, or so little that we can't measure it. That ends up being about 50,000 years. So carbon-14 dating is really good for measuring things that are less than 50,000 years old. Now, it's not going to tell us that something is 17,100 years old. It's going to tell us that it's about 17,000 years old. Okay? It's not exactly precise, except there are times when it is. This is where things get really cool. All right, so let's review before we go on. Okay, so let's say that you're a scientist, you found some bones. You measure the amount of C14, carbon-14 in the sample, and compare it to the amount of carbon-12. Now, we know how much carbon-14 and carbon-12 are in things of a certain age, and I'll get to that in just a moment. We know that the half-life is 5,700 years. So we use that ratio to determine how old the thing is, okay? If it has 12.5% of the carbon-14 that we would have expected it to have, then it's 17,000 years old, okay? Does that make sense? Right. Now, here's where things get really cool. Okay, wait, first, actually, let me do this part first, then I'll come back to that. All right, bristlecone pine trees. Bristlecone pine trees live in the White Mountains of California. There's a few other isolated populations at high altitudes in very, very dry environments. You can go to the protected bristlecone forest in the White Mountains in California. You have, if you've ever driven up to Mammoth, you drove through the Owens Valley, you looked left toward the Sierras, and you're like, ooh, those are so pretty and craggy and amazing. And then you look to the other side, and there were like these super brown, boring mountains, and you're like, that's boring. Look back to the pretty Sierras. The boring mountains, those were the White Mountains. If you drive up into the White Mountains, you would find the bristlecone forest. These trees are very cool because they live in a place where there's almost no water. It's very harsh, very high altitude, but because it's so dry, and such a harsh environment, there's also very few animals and almost no insects and fungus to break down dead organic matter. So these trees grow very, very slowly. The oldest that we found is about 5,000 years old. But the other thing is when they die and fall over, there's nothing to break them down. And so they stay on the ground for another several thousand years. So here's a cross-section of an ancient bristlecone pine that was alive at the time that Ramses II was ruling Egypt, okay, at 12,046 BC. You can see the tree rings here, and you know that trees put on one ring per year. In bristlecone pines, these tree rings are microscopic. Because they grow so very slowly, they put on very, very small rings each year. To really see the age of a bristlecone pine, you have to actually put it under a microscope. This is the science of dendrochronology. I know, coolest word ever. Dendro means tree, chrono means time, ology, of course, study. So it's tree time study. It's the science of determining the age by counting tree rings. You can determine not only the age of the trees, but also you can tell a lot about the environment the tree lived in by counting tree rings, because trees put on bigger rings when they have more water and resources. So you can tell when there were hot spells and dry spells and wet spells. So here's the other cool thing about the bristlecone pine trees. So they live so long that if we take carbon samples and measure the ratio of carbon-14 to carbon-12 in the different rings, we can find the exact ratio of carbon-14 to carbon-12 in living things that we know exactly how old they are. Because trees put on new growth every year, and the rings behind it are not alive anymore. They're dead, okay? They aren't still growing. So we can measure the carbon-14 back here, see the ratio, and figure out exactly how old it is. We can then compare that ratio to some other bone or piece of wood that we find and have a very precise estimate of the age. How cool is that? So here's a graphical description of that. So we've got the tree putting on bigger and smaller rings each year. Okay, so we take one tree that fell down. It's been lying there for a few thousand years. We compare it to a tree that's still standing up. So we can count the exact number of rings and tell exactly how old the tree is. Then we line up the matching rings, which we can do because the rings are different sizes each year. So here's our alive tree, okay? We compare that to a dead tree. We line up the matching rings, and now we can count back on that tree. And then we compare that to an older dead tree, line up matching rings, and count back from there. And so in this way, we have tissue samples. We know exactly how old they are. We can measure the exact amount of carbon-14 and carbon-12 in their tissues, calculate an exact ratio so that we know the exact amount of carbon-14 and 12 in tissues of specific ages. And when we find something that we don't know how old it is, we can compare it to these known samples and get a very accurate estimate of the age of that unknown sample. How cool is that? Because of the bristlecone pine trees, because they live so long, and then because they lie on the ground and don't rot, they have given us a very, very exact estimate of a record of carbon dating back about 30,000 years. How amazing is that? That's so cool. Okay, but carbon dating has some drawbacks. For one thing, it only works on live tissue. You can't use it to date rocks because they don't take in new carbon. So that doesn't work. The other thing is, every once in a while, we get some results that don't make sense. So here was a study that some people did on some clams. They carbon dated the clams that they had just taken out of a lake, so they knew they were alive. And the carbon dating showed that they were 1,000 years old. Well, we know they're not, because clams, like trees, put down new shell every year. You can count the rings on the clam, see how old the clam is. They're not 1,000 years old. Also, we just took them out of the river. We know they're not 1,000 years old. Scientists respond. These people said, we think what's happening is that these clams are in fact getting a lot of their carbon from the dead organic matter on the bottom of the lakes and rivers. So what we're seeing is old carbon that they've incorporated into their tissues. Now, here's how science works. They published this first article to say, this is what we got. Other scientists respond to that and criticize the original research and say, no, you're wrong. That's not what's happening here. Your study is flawed in this way and this way. We think what's happening is actually something different. The original authors can then respond back and say, no, we checked that. We're sure this is the mechanism that's going on. And this is one of the ways in which science is self-correcting, because scientists constantly argue and try to find out what's wrong with somebody else's research, okay? The idea that somebody publishes something and the rest of the scientific world goes, okay, cool, I will accept that at face value, is absolutely not what happens. So it turns out the carbon dating totally worked. These clams were living amongst really old carbon and somehow incorporating some of that carbon into their tissues. Turned out actually super cool. Carbon dating totally works. And in living things less than 30,000 years old, it's extremely accurate, in part because of the Coolio bristlecone pine trees. Okay, now let's go back to one other thing. This only works on things that were alive. So how do we figure out how old rocks are? This goes back to other isotopes. So if we go back all the way to here, these are carbon isotopes, but remember I said there were lots of other isotopes? We can use other naturally occurring isotopes. Uranium, for example. Uranium is naturally radioactive. Yeah, we use it in nuclear weapons and nuclear power plants. We can examine the amount of other things that have longer half-lives and were incorporated into the rock when the rock formed, and figure out how old those rocks are. So we call that radioisotope dating, okay, or radioisotopic dating. So carbon dating uses carbon isotopes. Other dating, either older things or things that are non-living, can be done using other radioactive isotopes. And that is the end of our basic chemistry. Next up, we're going to start talking about some of the biologically important molecules, how they form, and how your body uses them.